Let us now study about the applications of bioinformatics in the field of molecular medicine. Although scientists mapped the genomes of various organisms in the laboratories, the greatest challenge for them was the need to store or compile the huge biological data. For this purpose, computers had to be used in the research field as there was no other way for the storing of the data. Bioinformatics provided this opportunity to the researchers to store the data in the form of databases on computers. The whole work of DNA sequencing, its observation, analysis and interpretation all was the work of computers and it could not be done manually. The complete sequencing of the human genome has enabled to improve the biological research and clinical medicine. Scientists can find cures against diseases either hereditary such as cystic fibrosis and Huntington's disease or acquired as in the case of cancer or heart disease. Due to the complete knowledge of human genome, it is quite possible to view or analyze only those genes which are directly related with the disease. It is now easy to observe the molecular basis of each disease. This whole step will enable the pharmaceutical industry to design drugs which will only target those genes which are diseased. The drugs which are present in the market can target only 500 gene products or proteins, but the mapping of complete human genome has enabled to use the computational tools and more drug targets and medicines which could only interact with those proteins whose expression is diseased without any or little side effects. Pharmacogenomics, a branch of bioinformatics, has enabled to develop more effective clinical medicines. Though there are some advanced drugs in the market, but they have failed due to their adverse effects in patients because of the differences of DNA sequences in different patients. Today, as pharmacogenomics is making advances in the science world, doctors should use drugs on the trial and error basis because sometimes patients showing same symptoms of a particular disease show different results against the particular drug. It is because their DNA sequences are different. In the future, it will be possible for the scientist to prescribe drugs after observing the genetic profile of an individual. If the genetic mechanism of a disease is known, then it might be easy for the doctors to take diagnostic tests more accurately. There are chances that in the near future, it will be possible to use genes instead of medicines to cure particular disease. Gene therapy is one of the techniques which will provide this opportunity. In the future, it will be possible to cure or treat the disease by changing the expression of the diseased protein.